And the next speaker is André from Los Angeles. His title is What is the amount of drug uptake and release by nanocarriers at the tumor site? Optimism versus despair.
Well, thank you very much. So I'm going to say that my talk, as I intended it to deliver it, was given by Gert Storm yesterday. And so I was not going to repeat everything that he said. I think the whole story then involves about the controversy of Warren Chen's paper, and all I can say is that as the hype about nanomaterial delivery has receded, the real problems are becoming more apparent, but they are addressable, including consideration that delivery to the tumor site is determined by vascular access. Injected nanoparticles face physical and chemical barriers to overcome and can be overcome by rational design. So I'll show you a few of the techniques that we are currently used to improve that. Important to remember, drugs never go straight to the intended target. They reach targets as a result of properties that affect their stability in the circulation, water solubility, protein binding, and that needs PK, pharmacodynamics, and medicinal chemistry data. And we need to get past the idea that ligand-modified nanoparticles are magic bullets. While preserving the principles of PK, PD, and medicinal chemistry, nanocarriers can expand the idea of engineering past barriers, including the concepts embedded in EPR, which I don't think is a uniform principle, the tumor matrix, improving dosimetry, drug synergy, and multiple wave therapy. So this has been analyzed, and so I'm going to say very little in addition to the fact that the assertion was that only 0.7% of an injected nanoparticle dose ends up in a tumor, based on the study done by Wilhelm et al. With Warren Chen's lab. I personally just want to point out one thing that was perhaps not discussed as much yesterday. In order to calculate the area under the curve inside the tumor of this trapezoid, you basically have a formula that has the area under the curve multiplied by the mass of the tumor divided by T end. So if T end is very big, you're going to have a falsely low value or a value that is disproportionately low. And then if you have 117 different studies with many different types of nanomaterials where you didn't design T end, it could actually mean that many of the nanocarriers that was measured already deposited the drug load at the tumor site, and that you would then be using a time period that falsely reflect the integration. They also didn't measure drug delivery, and currently, whatever way you look at it, the value of a nanocarrier is assessed relevant to the reference value of the free drug. And FDA follows drug and not just carriers, but how the carrier biodistributes and release is important. So we all consider that. So I will not say much more about that study. So we've already heard this also very well covered in the meeting. How accurate is that assertion? Well, in both the fields of antibody and liposomal treatment for cancer, in mice and humans, delivery efficacy of the active ingredient generally ranges between 0.07 and 7%, and is effective in therapeutic outcomes. And then many different examples have been given also by Gert yesterday of saying that that calculation is actually not what we actually observe in studies. So I'll show you one study where we have a liposome equivalent nanomaterial, consists of a mesoporous silicon nanoparticle addressed by a lipid bilayer, and actually if the particle was designed with the fact that if you have this liposome lookalike, you actually have a particle with a more stable lipid bilayer, a huge interior loading space that gives you much higher drug loading capacity, and therefore less leakiness and increased retention and circulatory half-life. And that's borne out by the data where injecting irinotecan in this particle and comparing it to the irinotecan liposome, we get in the plasma, the payload equally improved as far as pharmacokinetics is concerned. When you actually get inside the tumor in a orthotopic tumor model, we achieve approximately a 5% ID with the silicosome compared to the liposome. And if you do the same experiment in a xenograft, the result is even better with a 10% retention, much better than the liposomal equivalent. It seemed to be stuck. And the efficacy in terms of treatment outcome then is if you compare the free irinotecan to the liposome versus the silicosome, a much better result with even animal survival in this study. So clearly, tumor biodistribution is an area that involves a lot of attention, and appropriately it was portrayed here that if you use companion diagnostics such as superparamagnetic nanoparticles as an imaging guide, the prediction can be made which patients are more likely to respond to nanotherapies. This was very well presented also in Omit's talk here. And then most recently, confirmation that ferromoxetol uptake in tumor lesions by MRI correlates with the response to the nanoliposomal irinotecan. This was published recently. So if we now get to the interesting question, what is EPR? Is EPR this effect of leaky blood vessels, or is it something else? Well, inherent in tumors is the fact that they need nutrition and that there is a transcytosis mechanism. And in fact, if you get into a cancer like pancreatic cancer, the fenestrations are covered by a dense stroma with parasites. So in a recent study from our group published last month in Journal of Clinical Investigation, we then used the silicosome, but we used one with an embedded gold particle in the core to actually look at this transcytosis mechanism. And so here is the tumor endothelial cell. This is the lumen. And if you now deliberately induce transcytosis by the RGD peptide, which functions via a receptor, binds to an integrin, and then gets cleaved to bind to the neurophilin receptor to drive the endocytosis pathway, we could very nicely demonstrate through transmission electron microscopy the induction of the transcytosis vesicles, being able to image the particles, being able to show that this quantitatively increases the amount of drug delivered to the tumor, and we could also follow the migration of these particles from the vascular lumen inside cancer cells undergoing apoptosis, where you can see the particles sitting in a perinuclear distribution. And moreover, as predicted by these MRI studies, if you compare patients now, where we had patient samples used in animal models, which have a high level of neurophilin expression on the blood vessels versus people with less neurophilin expression, there was clearly a difference in the effect of the RGD peptide, thereby saying that personalized medicine for drug distribution is important. Another way to think about the same thing in pancreatic cancer is where the endothelial cell adheres to the parasite. This mechanism is driven by many different factors, but including a major contribution by TGF-beta. If you use a nano construct, which is in this case a mesoporous silica particle with a polymer that binds through supramolecular chemistry, a TGF-beta inhibitor, you can actually move the parasites apart, and in a dramatic demonstration of a liposome being injected, very little of the liposome makes it into the tumor site, but if you prior deliver the TGF-beta inhibitor and follow within 1 hour, you can see a dramatic increase in the amount of particle uptake at the tumor site. You can also see that the liposome now distributes from a cortical distribution throughout the tumor site with increased efficacy of treatment. Finally, important to consider that drug synergy, that you may not need as much a drug if you make use of synergistic design, as for instance in Vioxx, where the cytarabine-duano-rubicin combination clearly can be calculated to show synergistic combinations if you ratiometric design them. We ourselves have been impressed by the fact that paclitaxel, the albumin nanosphere, is used to improve gemcitabine therapy by shrinking the stroma, and therefore did a synergistic design, making use of a software design to put just enough paclitaxel in the lipid bilayer to not have a cytotoxic effect, but being able to synergize with gemcitabine delivered from the particle interior to deliver a 9-fold increase in active gemcitabine, a 13-fold increase in the activated form of the drug that could be measured by HPLC at the tumor site, and a 4-fold reduction in the drug that is metabolized. And then if you use the ratiometric design principle, now you can also see that the efficacy of then comparing free gemcitabine to saline, you can see there's a big tumor. The tumor shrinks, but you still have tumor metastases. If you use, this is free gemcitabine. You shrink, but there's still tumor metastases. If you use the traditional Abraxane and gemcitabine combination, you also have tumor shrinkage, but still with metastases. The synergistic design particle here achieves total resolution of the tumor without the presence of metastases. So with that, I will conclude by saying that although passage to the tumor is important, there's a lot of ways that we can actually intervene to make current therapy more effective. Happy to take questions.
This is actually a question that also applies to the first speaker. And in your opinion, what do you think that is the best of among the different physical-chemical properties of nanoparticle? Which one is the most important in modulating the modality of entering the nanoparticle in a tumor mass?
Well, so I would say there isn't any one property that it is always a combination of property, but clearly the design of size is important in terms of the vascular transcytosis as well as potential EPR mechanisms. I think that the stability design of the particle and the materials that it is designed from to keep its cost of production low is very important. I think the zeta potential, or the charge on the particles, if you don't want a particle that is too cationic or too anionic, you want particles in the neutral zone. And then you can add to that a lot of smart design features if you now want to do additional things. But for every smart design feature you add cost.
Thank you.
More questions? If not, thank you.
Thank you.
And we go forward to the next one. Marco Sicardi from Liverpool, integrating physiochemical